Okay, so I heard you're thinking biochemistry and you want to know everything about amino acid. Well, stay here because this is your must summary video. Hi everybody, welcome back to the Premed Journal. I'm Maria and today I'm going to explain to you how amino acids work because I'm taking biochemistry and let me tell you, it's been a journey. But I actually like proteins and amino acids, so I'm going to explain it to you all. This video is going to be formed by three parts: the structure, the classification, and the key properties. And let's start with the structure. What do you need to form an amino acid? Well, you need a central key point, central carbon. You'll also see it in textbook as an alpha carbon. To it, you need a hydrogen. You also need a carboxyl group that is acidic and negatively charged and is marked COOH, and also an amino group which is basic and positively charged and it's written as NH2. Okay? To that you will have an R group or the radical group. This group will be different for each amino acid, and based on this group the amino acid can have different characteristics really, because we have a lot of amino acids and what differentiates one amino acid from the other is the radical group. 2. Classification. Okay, so here we can classify amino acids by two things: the R group or the necessity. But we're going to start with the R group. If we're going to categorize the amino acids by the radical group, we can have neutral amino acids that can be hydrophobic and non-polar, or hydrophobic means they don't like water, and also they can be hydrophobic, polar, they like water. Also, we can have here acidic amino acids. So remember we said to the mandatory structure we need a carboxyl group and an amino group. Okay, so the acidic amino acid had an extra carboxylic group, and then we can have basic amino acids, which means they have an extra amino group. If we are to categorize amino acids by necessity, we have essential amino acids, which means our body cannot produce these amino acids. And where do you need to find these amino acids? Well, by the diet. We need to have a balanced diet so we can take all of our vitamins, nutrients, and basically amino acids so we can survive and function properly. We have also non-essential amino acids. These are the amino acids that our body can produce by itself, but here's an extra category that is called conditional amino acids. So this basically is a group of amino acids that generally your body can produce, but in case of a deficiency or a condition or a sickness or something, you need to take these amino acids by, I don't know, medication, or to take it from food, or you need to take it from outside sources because your body cannot produce it by whatever cause. And now we're going to the last section, key properties. Key properties of the amino acid. One of them is isometry. What is isometry? Well, it means that our amino acid can be turned to the left or to the right. Imagine my hand. We have two forms. The D form, this is the right form, and the L form, this is the left form, okay? It's like a mirror effect. Imagine my hand, right? So my hand can turn like this. This is what the amino acid basically does. Imagine, this is the L form and this is the D form. It's the same hand, kind of, but they're switched. Yeah. But here's the trick, because an amino acid cannot produce proteins or linked to each other to form proteins if they are in the D form. They need to be in the L form. And here we have an exception. Glycine. Glycine is a neutral, is a non-chiral amino acid. Why? Because glycine does not have an radical group. What glycine has is an extra hydrogen, and I'm gonna put a picture so you kind of have an idea of it. Another characteristic that amino acids have is the amphoteric characteristic. So this is basically an auto-regulating characteristic. They act as a buffer based on the environment, and more specifically on the pH of the environment. And I'm gonna do a resume on the pH. We have the pH 7, which is the neutral pH. Everything below 7 is an acidic environment, and everything higher than 7 is a basic environment. So let's say we have an amino acid in an acidic environment. What is going to happen is that the amino group, which remember is basic, the amino group will receive a hydrogen, and we will not have NH2 anymore, we'll have NH3. Then, if we have an amino acid in a basic environment, the carboxylic group, which is the acidic group, will donate a hydrogen. Remember, amino group receives, carboxyl group gives. And also, when we have an amino acid in a basic environment and the carboxyl donates, we will get an amino acid that is positively charged. And when we have an amino acid in an acidic environment and the amino group receives, we will have an amino acid that is positively charged. And here we have a really interesting phenomenon. Here we have a zwitterion. Okay, so a zwitterion is basically when these two phenomena that I just explained happen at the same time. Okay, so at the same time the amino acid gets a hydrogen, and at the same time the carboxyl group gives a hydrogen. And you will ask me, okay, but what do we have at the end of this? Do we have an amino acid that is positively charged or negatively charged? Well, we'll obtain an amino acid that has the charge of 0. 0 charge. Okay guys, this is all I have for today. I hope I summarized this correctly and then you understood me. So yeah, this was the episode. I hope you liked it, and I will see you in the next one. Bye!